Jeff, you got something exciting. Tell us something exciting.
So I was watching from afar, so nice job, guys, on this Senate hearing. So actually, I was watching online, so there's actually a camera way behind you up in the corner of that hearing room, so I did listen in.
Was there shininess off his bald head?
No comments,
All right.
no, so I think it went great, so congratulations, pretty nice to have that hearing.
Yeah, thanks.
I agree. I thought it was very well done.
Cool. That was my announcement too. So take a step back, on Wednesday, we had a hearing in the Senate Ag Committee. It was on two bills, the bill to fund for evergreen research, the bill to fund continued grants or another round of grants to folks in the supply chain for evergreen crops. And we had an awesome slate of testifiers, including Alex. Don and I got to present, and Colin as well. And then we had a farmer, 3 or 4 businesses, Kearns Co-op. We had a retired Department of Health official who worked on drinking water protection. We had AURI, maybe one other or 2 others that I'm missing, but a really awesome slate of testifiers that I think landed very well with the committee. So, and we served them Kearns of cereal and Kearns of crackers. Next time we'll bring Hazel Snackers.
Well, next time bring enough to share with the audience. Who was very jealous.
I don't know if you can do that.
One other item as a heads up, it's apparent that the House Ag Committee and the Senate Ag Committee want to have a tour, a Forever Green tour, and it could very well happen during the first week of April. So we would think of giving them, a trip out to the field to see the green cover. We're pennycress out there along the road, and then the idea would be maybe a greenhouse tour and then a tour of food science. So if you guys have any other ideas, put them on the table in terms of what you think those legislators in their age should see when they come to the St. Paul campus. All right.
This is Randy Ellingbo, by the way. I thought it was a great hearing. Nice job on the presentations and answering the questions. And yeah, in regard to the tour, certainly giving them samples was a very nice idea. They enjoyed that.
There was only one issue. There was one person that went long. We won't say who it was. You know what I mean?
Yeah.
We'll get that individual straightened out as soon as we can.
2 years. So Randy, who just spoke up, if for folks who don't know, was one of our testifiers, speaking from many years of experience in drinking water protection, and that was an awesome addition to our slate of testifiers. So thanks, Randy.
It was nice to be asked. Thank you.
All right, any other announcements, legislative or otherwise? Okay, I will hand it over to Lois to run the show here.
Okay. So. Am I supposed to read this?
Nope,
No,
you can just
just.
go on to your,
Okay. Oh, wait, no, there.
yeah, okay, good. That's what.
Yeah, okay. So this is the first of 2 sessions on hazelnuts. Today, I'm going to start with the introduction, hazelnut breeding and agronomics. Because of time, I don't think we'll get to very much agronomics, and I don't really have that much new on agronomics. But Scott Brainard and Julie Dawson are colleagues in the hazelnut program based in at UW-Madison. Julie's a plant breeder and Scott is her is a postdoc. Julie was going to present, but she has a scheduling conflict, so Scott's going to present for her. Scott has a few slides in the middle of my presentation, so we're gonna toggle back and forth between us. And then when we finish, Devin Peterson, who is a food scientist, who was formerly here at the U of M, but is now at Ohio State, will present.
Devin,
How you doing, Don?
fine.
Good to see you. Good, and thanks for including me in the meeting, Lois and team.
I keep up and know a lot about Devin through his mother-in-law that lives 500 feet from my house, so.
Yeah. I'd love to hear about those conversations sometime.
I tried to figure out a way to get this presentation so that Scott could go first, because Scott had his birthday yesterday, so happy birthday, Scott. And Scott is anxious to get on the road so he can go celebrate. Whoops, I think I doubled. So, actually, I think I've already kind of covered this outline. So I'm going to start with the background, first generation selections, second generation selections, and then our newest controlled crosses. And then Scott's going to talk about genomic tools, which Julie was going to have covered. And then Devin is going to talk about tools for evaluating flavor, which was covered with, is kind of under the rubric of developing advanced tools for breeding. So most of the people in this audience, I don't know who all is on Zoom, probably already know that these are hybrids between American hazelnut and European hazelnut. The European hazelnuts having been developed for commercial production with large nuts, thin shells, whereas they're highly disease susceptible and not hardy in the upper Midwest. The American hazelnuts are just the opposite. They're disease resistant, winter hardy, but small nuts and thick shells. So not considered commercially viable, but that would be a matter of discussion later on. Just showing you a picture of what the Eastern filbert blight looks like. On the left, there are cankers that basically choke off the vascular system of the hazelnuts and cause branch death, which you can see in the bush on the right, which actually is not a Midwest hybrid hazelnut and was highly susceptible to Eastern filbert blight. But the bush in the middle shows some cankers that I identified in 2008. And as of 2020, that bush still looked mostly fine. So that shows the level of tolerance that our germplasm has for the disease. So here's a comparison of, on the left, we've got, well, actually, on the bottom, we've got Tonta di Giffoni, which is a European selection that is considered the standard that the Ferrero candy company that makes Nutella wants hazelnuts to meet their standards in terms of roundness and size and degree of skin on the kernel. If you look above, those are a pure American selection identified in Wisconsin, much smaller nuts, but you can see they're also pretty round, and they actually have a much lower quantity of skin on the kernel. And according to people who study flavor, they actually have excellent flavor. So anyway, our hybrids are somewhere in between those two in terms of size. So our selections are winter hardy. That's actually pretty easy. You just plant them out in the field, and if they survive, we assume they're hardy. Eastern filbert blight resistance, again, we inoculate our seedlings. If they don't have Eastern filbert blight after 5 years, they're probably resistant. So what we're focusing on is consistently high kernel yield, higher percentage kernel, and kernel yield is the multiplication of in-shell kernel size or kernel yield. I mean, in-shell yield multiplied by shell out rate, the percentage of the total shell that is kernel. The Americana typically have less than a third of the total nut weight is kernel. And therefore, you harvest a lot of tonnage of nuts, but get very little kernel for it. So we multiplied the two together to get kernel yield, and that's actually the primary thing that we're selecting on. And then we're also selecting on acceptable kernel quality. We're looking for slightly larger nuts, lack of fiber, and so forth. And some of these things are subjective, and we're still discussing how much we need of them. We're also selecting, we would like shrubs that are conducive to machine harvest, and we're still trying to define exactly what that looks like, because the European hazelnuts are actually grown as trees in Oregon. Our bushes are shrubby and multi-stemmed, and when they're heavily laden, they kind of flop over. But that might actually be just fine for machine harvest.
Some of them, well,
Yeah, some of them do. So we're also looking at insect and other arthropod resistance. That's early in the work, and outstanding flavor, which is why Devin's part of our team. So the breeding cycle of hazelnuts in Oregon is 17 years. Basically, they do their crosses, they plant out the seedlings, and evaluate them. You know, they take about 3-4 years before they come into nut production. They evaluate them for 3 years to identify the best bushes, and then they clone them and put them into replicated trials to evaluate yield under controlled conditions, and do that for another 7 or 8 years. And it all adds up to 17 years of cycle. We started in the middle of that cycle by working with on-farm growers, of which there were quite a few already, to identify their best plants, because we had all these populations of plantings on farms that were all propagated through seed. So we started in the middle of the cycle, and these are some of the people who helped us identify the best plants. We put them into replicated trials on campus, starting in spring 2009, and by about 2013-14, we were getting enough yield data that we started to identify the top selections. This picture was what we thought were top selections in 2019. We keep on refining that list, so some, you can see the stars at the bottom of the diagram there. The ones that we're trying to move forward are those that we continue to, well, should I say the ones that don't have stars are ones that we've since discovered things that we don't like about them. For example, this Brainard 342 has ESD. It probably has tolerance to it, but it does seem to be dragging down its yield potential, because I haven't seen much yield on it lately. FPC 2D5 has big bud mite on it. Is it a problem? Maybe not in the long run. We're still studying it, but we're not moving it forward in the meantime. But all the others on this, well, Shepherd also is a European type hazelnut, which we initially selected because it had large nuts and thin shells, but it turns out it really doesn't yield very much. So we've since been less impressed with it and dropped it on our list, but we're trying to move all the rest forward. And some of them were moving forward faster because we are actually more successful with propagation with them. And propagation is the key, is the limiting factor. So you see the two that have double stars underneath them are 2 of the 5 that we have been able to propagate and are actually seeking to get IP on now. So it wasn't until we were able to have enough plants of the same genotype to pool their crops and put them in through the processing equipment that we were really able to see that, yes, there are genotypic distinctively different nuts. So these 3 are 3 of the 5 that we're moving forward. And you can see that 9-2 has darker nuts, and that's because it has this skin on it, this fibrous stuff on the outside of the kernel that's undesirable because, yeah, it doesn't really feel that good in your mouth. But it turns out that after you roast a nut, it rubs off very easily, so it's not really a problem. So our next step is to propagate these again for even more replicated trials. Our first round of replicated trials included 149 selections at 5 locations. Now we're having a smaller number of selections. It's about 18 or 19 that we're putting in small trials on farms in many, many different locations to, fine tune it, say, are these widely adapted or just narrowly adapted. So these trials have gone into 14 on-farm trials in Minnesota, Wisconsin, and Iowa. 10 joint performance trials, and these are trials where we're comparing them with our top selections from other breeding programs in the Eastern United States, including New Jersey and Grimmel Nut Nursery in Ontario, and then 2 selections identified in Nebraska. So we've got 2 of these trials in Minnesota, 2 in Wisconsin, 2 in Iowa, and we're also sending our selections back to people who've contributed plant start plant material. So, for example, there's also our material in New Jersey and in Ontario, and we also have collaborators in Nebraska and Quebec, and I keep on getting requests from even further afield. And so we're also putting our new selections into agronomic trials because now is our opportunity to address questions about fertilization. That when I did my graduate work working on fertilization with seedlings, I found the seedlings were just so variable that they obscured the response to fertilization. Plant spacing is also a big issue. In Oregon, they grow the hazelnuts as trees, 10 feet apart. That's probably not necessary for our material. We can squeeze it in further, but we want to know how much should we squeeze it in before competition between plants reduces yield and shading and so forth. Is pruning useful? That kind of question. So we're establishing trials for those. And then we're also hoping, starting soon, to identify larger scale commercial plantings that were called go first farms to actually commercialize them. And for those of you who attended the hearing on Wednesday, there's this valley of death in commercialization that we need to get across by probably subsidizing these farms to de-risk the commercialization. So a few results from the joint performance trials. One of them's here in St. Paul. Unfortunately, we got 0 nuts from the trial in St. Paul this year. Probably because it was 2 years in a row of drought, and combined with the fact that we've probably got the worst land on the whole St. Paul campus. It's on this rocky, eroded hillside. For those who are concerned, I've got money now to install drip irrigation there because it was beyond the reach of the sprinkler system. So we couldn't really irrigate it effectively. So hopefully we will start getting yield from that trial this year. But Jason, my colleague down in Wisconsin, did get yield from the Madison trial. And this is, I had a graph, but I decided I like this kind of physical graph much better. So the 6 bags on the left are Northern Blaze, which is a selection from Grimmel Nut Nursery in Ontario. And it just keeps on blowing everything else out of the water in terms of productivity. The next one, Marion, is also from Braun Nut Nursery. I should back up a little bit and say that unfortunately, we've got an uneven stand establishment date for their different selections in their joint performance trials. The Grimmel sent us plans in 2017, when we said, wait a second, we haven't figured out how to propagate our material yet. So the oldest plants of our Midwest selections that are in those trials are 2 years younger. And I think the Grimos also sent us material that was already 2 years old. So it's been producing, and our material is just now catching up. So the third one there, ARB 7-1, is one of our plants, and you'll see that it was established in 2019. Everything else is planted in 2017. So it's third in the ranking, even though it's 2 years younger. The next two plants, Grand Traverse and the Beast, are two that were identified by the Hybrid Hazelnut Consortium, which is Rutgers University, University of Nebraska, now Center for Agroforestry in Missouri, and Oregon State University. So they've been pushing Grand Traverse and the Beast. I can tell you definitively, Grand Traverse and the Beast get knocked back by winter every year here in Minnesota. So don't even think of those two plants here in Minnesota. But they seem to be doing all right at Madison, which is slightly further south. But not nearly as well as Northern Blaze. So we're actually currently in discussion with Grimo Nut Nursery about licensing Northern Blaze for distribution in the upper Midwest. They have been very generous, and we want to make sure that they get what is due to them in terms of ownership of that selection. So here are some pictures, some closer up pictures of those kernels, and you can see the 4 Grimo selections do indeed produce very nice, beautiful nuts. And our nuts by comparison, our kernels by comparison are smaller. Rose 9-2, as previously mentioned, has a lot of skin. Our 4-3 actually has very, very nice nuts from a culinary perspective. Now I think it's Scott's turn, so I need to let Scott speak. I hope Scott is
Thanks.
on.
Thanks, Lois. Can you hear me?
Yeah.
Great. So yeah, I have a few slides sprinkled into Lois's presentation here about work that's going on at the Spring Green, Wisconsin location of the Savannah Institute's collaboration and assistance with this stage of the kind of scale up of some of these varieties. And then I'll speak later, longer after Lois's presentation about some of the breeding work that we're doing, that I'm doing in Julie Dawson's lab at the University of Wisconsin.
Give a little better introduction of yourself, if you might.
Sure. Yeah. So I'm a postdoctoral researcher in Julie Dawson's lab at the Department of Horticulture at the University of Wisconsin. I was trained as a plant breeder and joined Julie's group specifically in the context of this USDA SCREE grant, where my task is to develop genomic tools to make breeding for a variety of traits more efficient using essentially using next generation sequencing methods. So I'll say a bunch more about that, the motivation and the context in a bit. But for now, just to give some additional kind of context and color to what Lois is presenting, this is a drone shot of one of the farm locations in Spring Green, where again, in collaboration with this nonprofit organization, Savannah Institute, we've planted what we're calling one of these Go First farms that's testing out a few of the UMHDI varieties along with Grand Traverse and the Beast, in order to assess its, at a larger scale, what the commercial viability of these clones would look like, what their yield density is. And in particular, as I've laid out here, this is also a bit of a overlaid on top of that kind of commercialization angle is a research question of specifically at this location, a density trial. So figuring out what the sort of optimum space in row, so between plant spacing of these UMHDI plants is. balancing or sort of optimizing that question of plant cost versus eventual yield and sort of time to canopy closure and all of those things. So we've got 3 different spacings of the varieties randomized in 3 different blocks here. And then at a different location, we've also planted these clones in order to assess different pruning management systems and again, how that affects all sorts of things, sort of the full suite of mechanical management, mechanical harvest, as well as yield. So these include, Lois, if you want to advance one slide, I think I have one more that may be a bit hard to read. This is the field map for the location I just mentioned, but sort of illustrates that we've got, if you can read this, a number of the UMHDI varieties here, including ARB 7.1, Rose 9.2, Price W41, State Bend 7.6, along with Grand Traverse and the Beast. And there's also some Grimo varieties in the mix here. So, and we hope to expand this with additional varieties as they become available so that this can be not a rigorous, sort of variety trial location, but be a place where advanced clonal selections that have sort of already been validated as worthwhile to investigate for like scaled commercial production can be stacked up against one another. So we don't just see what does ARB 7.1 do when you grow 50 feet of it or 150 feet of it at a specific spacing, but we can directly compare that in a similar climate on a similar soil type to many of these other varieties. So I think that's it, Lois. I'll turn it back over to you. And then I think I have another little snippet to say later. And then, yeah, thanks.
Can you say anything more about the intercrops?
Sure. Yeah. It's there. It's out there on the field map. So we're growing vegetables in between these rows, essentially as a way to, I mean, again, this is a kind of pilot farm, and we are trying to pilot different cash flow options for farmers who are who in reality would need to cash flow the establishment years of these perennial crops prior to them actually bearing a marketable nut yield. With, in this case, we're growing vegetables, so annual root crops, and tracking the financial aspect of that is one of the jobs of the farmer at Savannah Institute, Eric Hagan, who's taken the lead on sort of designing this particular system, and hopefully having this not be something where we just get data that we can report back about the yield density per square centimeter of canopy cover, but also bring farmers and prospective early adopters and say, this is a system that actually worked for us in a real world scenario. We're growing it on the hilly topography of the driftless in southwestern Wisconsin, and so all the challenges that come along with that are being represented here in hopefully a realistic way.
I love it. Yeah.
Thanks.
It takes 3-4 years before you harvest any yield from those hazelnuts. So
yeah, well, they had 1 or 2 nuts on them in their first year, but you can't living off of that.
age pregnancies.
Yep.
So, okay, so moving on to the second generation, and these are from controlled crosses that we made from 2014. Well, I'm focusing on 2014 and 15. This was really early in our identification of the top selections. So not all of the plants, not all the crosses that we made, not all the parents of the crosses that we made turned out to be ones that we ultimately selected, but we lucked out in a few cases. So our biggest planting of those is at Rosemount. We've got 7 acres there. And this aerial photograph shows quite distinctively the differences between those crosses that we made between our top Midwest selections and using pollen from Oregon State University. And those are the larger statured plants that are actually most of the field and that are, yeah, the European, exactly, the Oregon pollen, pure European. We also made a few crosses that were our top selections times our top selections. So they're Midwest by Midwest. And you see in the lower left corner, the redder colored plants, the shorter statured plants are the Midwest by Midwest. And they actually have much more beautiful autumn leaf color than the European, which desirable for people wanting to grow them as ornamentals.
No nuts, but they're pretty.
Actually, they
That's
are
the
heavily,
exact opposite of that, actually.
they're actually very heavily laden. So, last time I gave this presentation, Dawn asked about EFB resistance. And so I spent a lot of time putting together this table based on the large families that we had, comparing what's known about the EFB resistance of the pollen parents and the EFB resistance of the maternal parents. I'm not going to call any of our maternal parents resistant so much as tolerant, but the one on the bottom, plant B2-7 and Gibbs 5-15, I've never observed EFB on either of those plants. As you go up the table, I've seen higher and higher levels of EFB on the maternal plant. And likewise for the pollen parent, on the left is gas-away resistance, single gene resistance, which is what they're primarily relying on for resistance in Oregon. And then in the middle is Grand Traverse resistance, which is quantitative resistance from this Grand Traverse selection that is being promoted by Rutgers University as a potential variety for commercial use. And then two numbered selections from Oregon State with quantitative resistance, and then the one on the far right with an unknown, which is basically put in there for a control. And although it looks like the gas-away resistance is sort of holding up here, it's not complete. Likewise, the quantitative resistance, you see one there
You want to
that
Oregon's quantitative, not.
exactly. Oregon's quantitative. So anyway, the take-home message from this is that it's a good idea to have resistance from both parents. And but that also the patterns are a little perplexing. And this is basically the same information in a different format. And if you look at the upper left graph, it shows that assumed resistance by on Midwest plant by the gas-away resistance gene on our side, it's getting a high level of resistance in our progeny. Whereas susceptible by susceptible in the upper right, or the lower right, I mean, we've got much lower levels. So anyway. So the primary reason for making these crosses was to improve nut quality. And you can see that, yes, the crosses with Oregon State's material, the progeny are in fact much larger. But how that affects yield is another question. So here's Scott.
Okay. Yes. Thanks. Yeah. So this is just almost a prelude to what I'll discuss a little bit more in detail in a moment, but I wanted to throw this in here as just a specifically like discussion of a key phenotype that we're using as a early as a trait to look at as a sort of test of some of these genomic methods that could accelerate breeding, and that is physical characteristics of nut morphology, which are relatively easy to evaluate, show in previous research to have smaller environmental variance components, so amenable to unreplicated trials where we're dealing with a lot of seedling material, and also like obviously very important, I mean, both from a nut quality perspective in terms of the shape and size of the kernel, but also, as Lois was alluding to, percentage kernel being a direct yield component in a crop like hazelnut, where kernel fill is very important. So just about how we're actually doing this in the Dawson group is we are using digital image analysis to just sort of speed the whole process along and gain a little bit more precision in our ability to measure these traits quantitatively, which is really essential from a genetic analysis perspective. We take an overhead shot of a 30 nut, ideally sometimes fewer, subsample of nuts from a given bush in shell, and then we crack them out and put the kernels back in those same squares and take a photo of the kernels. And that way we're able to just basically instantaneously with a few lines of computer code, associate kernels with nuts, and not only phenotype the nuts themselves in shell and the kernels themselves cracked out, but get measures of percent kernel directly. Yeah, so this is the glamorous life of especially an undergraduate in a lab who there's been a lot of hours spent taking photos of nuts. But we get to QR code everything, we have a permanent record, and in my experience, this is a much less error-prone way of trying to collect a lot of data. So if you want to advance the slide, Lois, I think I have some pictures of how this happens. Yeah, so we crop out each box and use image detection methods to actually identify the kernel and the in-shell nut. And then we take that and create it's called a binary mask. So we just can basically we can extract that portion of the image and measure like things like sphericity, sort of. I mean, we can measure the circularness of this nut and add in a z-axis measurement that we make by hand with calipers to get an approximation of how spherical the nut is, as well as its length and its width. And we also do bulk measurements of weight while we're doing this whole process. So if you want to advance one more slide. These are some histograms of kernel traits where I think this is just interesting. I mean, it gives you a sense of the kind of data you get out of this, but also just a interesting visualization of a difference in the populations we're studying. We were studying 3 F1 families that Lois's group made by crossing UMHDI maternal parents with pollen from Oregon State, pure avellana. That's what's shown here in the sort of teal color. And in the reddish hue is a population of Corylus americana sourced from the Department of Natural Resources, growing in Wisconsin, just on a farmer's field. And my comment here would be that there's obviously more diversity or more variance in the Minnesota families, which should be expected in a like true F1 population. But also you can see the general phenomenon here kind of laid out clearly quantitatively, that the nuts off of even an F1 hybrid with avellana are larger by every measure than the what we are considering here to be pure Americana. But the sphericity, the shape of the nut, which is shown in the lower right, was the length width ratio, are actually essentially equivalent. A length width ratio of 1 would be a perfect circle. So if you want to advance another slide, these will be the same. You can see similar things with the in-shell traits, but here some kind of composite measures of percent kernel, both volumetrically and gravimetrically, so from the images or from mass. And again, the percent kernel variance is higher in general, although it's not as striking with the Minnesota families. But in particular, from the weight perspective, there's the means of these two populations are not that different. And so this is, again, sort of general theme, I would say, being borne out in the data, that the nuts off of Corylus americana are smaller, but they are not necessarily yielding any less kernel per acre once you take into account that smaller kernel size and the smaller stature of the bushes. Again, the lower left, nuts per cluster. There are more nuts in a given cluster, in an average cluster from Corylus americana, and they're smaller. And so it all, like, the idea is that maybe this all comes out in the wash, and these are actually extremely competitive from a yield perspective. And I think that's it. There may be, I forget if there's one more slide, if I sent you another slide or if I left it in for my other. No, okay, so yes, that's all for me on nut morphology for now.
Okay. Thank you. So, perfect. So whereas Scott is looking at the genomics, my team is primarily focused on just finding the good plants, regardless of what the parentage is. And we've now got a list of 32 plants that we have identified that we are going to propagate next year and move forward into the replicated yield trials. And the new Forever Green grant that we just received is specifically for funding for that. Unfortunately, I didn't have room on the slide to present all 30 of them, but this is the top, these are the top 20. And what I wanted to note is that although, so how these are ranked, I spent a lot of time playing around with it and saying, what traits are most important? And changing my mind about it as well. So what I ranked these on finally, so we've got kernel percent multiplied by in-shell yield comes out with the kernel yield. And then we divide that by the area of the plant, the canopy area of the plant to get yield efficiency. It's a little bit debatable about, well, even if the plants are smaller, can you really crowd them in more in the field? And that will depend on what machinery we end up using. So that's not completely resolved question. But for the time being, I'm using yield efficiency and then multiplying it by kernel weight to get a score that I'm calling harvest index or yield index, whatever, and ranking them according to that. So these are the top plants. I did fudge a few things at the bottom of the list of 32 plants to get a little bit more diversity in the selections that we're going to select. It turns out that as I played around with different weighting different traits differently, the plants at the top of the score list, they shuffled within the rankings, but they were generally the same. It's the ones that were down below number 20 to 32 that moved around more radically. So I'm confident of these 20, that these definitely need to be looked at.
How does that top yield compare to Oregon?
Good question. I don't remember off the top of my head, but I can look it up.
That plant this year yielded the per acre equivalent. You assume that the plant will cover at maturity 50% of the surface area looking from above of an acre, yielded the equivalent of 1,400 pounds of kernel.
Compared to
That's
Oregon.
That's Oregon.
so these are in range or touching of the yields out of Oregon.
Single bush rep 1 year, caveats, but yeah,
Yeah,
they're in the neighborhood.
they're in the neighborhood.
What is the background, the Lamb W's that Midwest selection?
Lamb W is the Midwest selection. So I'd like you to note, those that are shaded pink are all the same family, Eric 4-21 times Jefferson. Jefferson also appears a lot up there. Jefferson has a single gene gas away resistance. The ones shaded in orange are Midwest by Midwest. So I just wanted you to note that there are 2 of them in the top 20, and then there are additional ones in the top.
But based on the last presentation of the Americana by Americana, where would those best lines reside in this list?
This list doesn't have any of those in there.
Do we know those yields? From
I could get that
from the other,
information from Scott.
there was a lot of Americana work
Yeah,
done in the initial backing of this program.
yeah,
I'd love to see, based on what he just described to us.
Right.
How does the Americana by Americana compare to what you were laying out here? I would suggest that be rebuilt back into the program.
Mhm.
So this is.
Is it line 12, a Midwest by Midwest?
Yeah,
And that's-
not Americana.
Yeah, it's-
Oh.
the Midwest are.
And it's specific,
hybrids.
in Pacific.
Oh.
Yeah, they're hybrids that have been going through recurrent selection for many generations.
So when that's what I
Okay.
want to make sure. that
So
got-
Midwest and Americana are different things.
Correct.
I knew that. I know that.
Generally,
word
what we call it, the Midwest hybrids that come out of the original round of performance trial selections much more resemble the American hazelnut. And the European hazelnut, and then also what most of the plants in this field look like. And this is not exactly Dawn's question, but something to note is that the way that these are ranked by multiplying kernel yield and kernel percent by the kernel yield efficiency penalizes the Midwest by Midwest crosses, because they have much smaller nuts. So the kernel yield of them is higher than their ranking ends up being.
Question over here?
Um,
No.
you might have said this, and maybe Scott has, are you selecting for like shellability? Is there variation for shellability?
That's basically, well, one of the things I was thinking about with Scott's slides is you saw that he did, percent kernel volumetrically versus percent kernel gravimetrically. And although we like higher percent kernel, because for however much weight you haul out of the field, you get more kernels that way.
Yeah.
But it turns out that shellability also has to do with how much airspace there is.
So what's
Mm.
gravimetric?
Gravimetrically is when you weigh a sample and crack it out and then weigh the kernel. Whereas volumetric is you actually measure the volume, which is what Scott's done. We don't have.
Having a little
way to
gap
do that.
space is good.
To have a
for-
little airspace in there, because when you crack the nut,
That makes
it's
sense.
pressed, and if there's airspace, you're less likely to crack split the kernel. And what I find with our Midwest hybrids is a lot of times they're more tightly pressed
Yeah.
against the shell than the European or the background, the F1s are.
Mhm.
And so they're more likely to have splits, but I actually have a slide about that. So shellability is in fact a very big issue, but that might actually be an issue to be solved by the engineers. And so,
I can ask another question or do you have more slides?
yeah, I'm gonna skip ahead. These are just pretty pictures. I'm skipping ahead because I want to make sure, and I've already talked about this. So anyway, we are basically reconsidering the nut quality issues about what exactly are we looking for. Thinking, I made that point about the Ferrero ideal nut. Well, we're not interested, at least most of the people I work with aren't really interested in feeding into Ferrero and their market and being beholden to whatever price they want to pay us. We're thinking more in terms of developing our own markets. How realistic that is, I'll let the commercialization people think about that. But we're thinking that our smaller nuts might be just fine. And, oh, I should have put this picture earlier. So this shows how the smaller Midwest by Midwest ones actually bear their nuts on the outside of the branches. So they're actually much more visibly heavily laden, whereas the European ones have the nuts, larger nuts, but fewer nuts per cluster on the internal of the branches. So when the harvesting equipment comes through, sometimes it might not work as well for our the top harvesting system as the Midwest by Midwest. We don't have enough plants of these new crosses out there to machine harvest them yet. So we don't know the answer to that question yet, but it's something to consider. So here's a comparison. It's not a completely fair comparison because we didn't harvest every single plant of every single family out there. We only harvested the ones that stood out as being exceptionally good. But in most parameters, the Midwest by Oregon State look to be better, except when it comes down to yield efficiency, where actually over the course of time, we actually have higher yield efficiency with the Midwest West project. So, and then here's the slide that, darn. Well, I might actually be able to.
The old lateness side.
Oh, I suppose I can't fix that. This shows you a new piece of equipment that Jason Fishbach and Ashland just got, which can sort out, it's called the color sorter, and it can sort out a mixture of shells and split hazelnuts mechanically, really quickly. And the top picture that got cut off there is the split kernels that came out of the batch that at the bottom picture, you've got a bunch of shells. Darn, I should have noticed that and fixed that earlier.
Yeah, so these were mixed together before, and the top photo has visibly that top layer of it has like 3 shell fragments in it.
Right.
It's-
So this might.
So it'd be just a small lawsuit, right?
No,
Fewer lawsuits.
maybe just grind it, grind it, oil and meal.
There would
It has a
still
little
be
bit of
people
shell.
hand picking probably, but when there are only a few shells, it's very quick. Most nuts industries still have hand pickers.
Is your flavor guy gonna make a presentation?
Yeah, and I need to hurry here, so I've just got,
Yes, it's happening.
Did more
I'm
crosses.
gonna in and out today.
and I think we can skip all that and we're gonna go over to Scott.
We need to see the presenters now.
Yes.
Scott or Devin?
Great. I think you have to stop sharing for me to start. Lois.
You want to ask them if you use them as a urgent time thing.
Yeah, does anybody need to go? We're at the end of the hour. This is an hour and a half meeting, but if anybody needs to go quicker, we can prioritize somebody.
Also, yeah, just a little time check, I guess. This is about a 10-minute presentation. Is that still?
You're
looking-
fine, you're fine.
Okay. And you can all see this, I hope?
Yes.
Okay, great. So yeah, I've already, I think, said who I am and what I'm doing. This is some of my collaborators here, in addition to Lois, my advisor, Julie, of course, Jason, who's with UMHDI and UW Wisconsin, and Dean Sanders, who's at the Biotechnology Center here at UW-Madison, have all helped a lot with this project. So what I'd like to do is just quickly explain in more specific detail why we're doing this, and then I've already kind of introduced the phenotypes, so I'll move on and just discuss one aspect of the results we've gotten so far, and conclude with some notes on where we're going from here. So Lois has already explained this, I think, in sufficient detail. We are trying to interspecifically hybridize Corylus americana with Avellana, either, not by making F1s necessarily these days, but this is the background for a lot of the breeding that's been going on for decades. And the idea is to combine high value, high yielding nut varieties from Avellana with the biotic and abiotic resistances from Americana. And these are some of the current results. Okay, so why should we try to integrate genomic data into the selection process these days? And I'd say the short answer to that is it's taken a long time to get to where we are right now. And if there's anything we can do to speed that process up in the future, it's worth investing in and looking into, because the barrier to entry for a lot of these genomic technologies has dropped significantly as the cost of sequencing itself has plummeted. So as Lois has explained, her program has been based on the phenotypic evaluation of individual plants. So phenotypic selection is very resource intensive. You have to wait years for traits to express themselves. You have to score it by hand. And that entire time you have to be paying for land, you have to be paying for management. And it's, in a perennial crop in particular, I mean, this is true, I would say, across every crop species, but as the generation time and the time to maturity gets longer, this resource investment in phenotypic selection gets more and more intense. Genotypic information can be obtained when the plant is in its, has just produced a leaf in the greenhouse still, very little investment at that stage. And it costs, again, a ever decreasing amount of money. So our goal is to try to use this genotypic information to select better parents for crossing and to screen larger families while they're still in the greenhouse, and thereby increase selection intensity. If we can guess what the performance of a plant will look like 10 years from now using just its genetic information, even if that's not a perfect guess, it still can have significant benefits for the speed with which you make progress. So like I said, we're interested in lots of traits in principle, but we're starting with this trait as an example that we think would be easier than some more complex traits. So this is both something we could do right away, but also is maybe a bit of a bellwether for the potential utility of these methods, since it probably won't get any easier, trying to predict traits like yield, for instance. I didn't show this slide, which is sort of this is picking up where I left off, essentially. This is a somewhat complicated plot, and the only point I wanted to make here is that I alluded to a lot of different traits that you can measure using these digital images. And one question you might have is how correlated they are with each other and in what direction, positively or negatively. And the upshot here is that all these arrows pointing to the right, kernel width, kernel weight, nut length, which you can't even necessarily read the labels on all these arrows, are positively correlated with each other, which makes sense. A bigger nut probably contains a bigger kernel, and it probably weighs more. But the good news here is that when these arrows that are pointed at 90 degrees from each other indicate, in this particular visualization, which is called principal components analysis, traits that are not correlated with each other. So in particular, if you look here, the gravimetric kernel percentage, so what percentage by weight is that nut composed of kernel, is not correlated with the size. And again, that was almost evident from the histograms I showed before, but this is just showing in a different way. It means that you can select for both of those traits at the same time without suffering penalty, in the sense that if they are negatively correlated, that might not be a statistical aberration. That might be that they are like linked genetically in opposite directions. So that's some good news about our ability to make gains on multiple traits at the same time. That, yeah, we don't need a big kernel in order to have high percent kernel. Okay, so there are a lot of different ways of using, that's the phenotypic data. The genotypic data we get by genotyping a whole bunch of bushes using, there's a lot of different methods, and I'm not going to get into them, but what you obtain are polymorphisms, genetic polymorphisms across a population. And so you have to choose a population that you want to study, where you collect both phenotypic data and genotypic data on the same bushes. In particular, I want to talk here about some F1 populations. So these are controlled crosses where known specific parents were crossed together. And I hope I have these dates right, Lois, actually. I should have checked with you. But this is a sort of general scheme for what an F1 looks like. You make a controlled cross, and then, of course, you have more than 8 progeny. We have about 100 bushes per individual. The color scheme is just here meant to represent that when you do this, you generate recombination. This is what happens during meiosis. And so both parental individuals here generate gametes that represent a recombination of their genome, and they come together in the progeny. And using statistical methods, we can actually figure out where those recombination breaks happened, and then attempt to associate specific regions of the genome with variants in the phenotypic traits that we're collecting. These are called QTL, quantitative trait loci, that often contain multiple genes, but the theory is that some or all of those genes are contributing to the variants in the trait that you're observing. So this is one of the ways that you could then think about using genetic information to use in a selection program. It's not the only way, and I'll make some mention of other methods that we're exploring at the end. But I do want to just talk about this population in particular now, again, sort of as an example. This is one that was planted, I believe, in 2014 and 2015. The parents, though, I believe I am 100% correct that the paternal parent was this Corylus avellana selection from Oregon State called Jefferson, and the mom was Eric 421. The markers.
They were planted in 2015. They were the crosses were made in 2014.
Thank you, Lois. I always get that confused. So again, you can use lots of different ways of genotyping plants these days. There's, a plethora of options. We used what's called Illumina sequencing. It's a short read sequencer. So you chop the genome up into tiny little pieces and you sequence those tiny pieces many, many times in order to get it right. We then, and then there's lots of different polymorphisms you might look for. We looked for insertion deletion polymorphisms, and we identified those using this bioinformatic tool called STAX. And we used also a new reference genome assembly for an old important Corylus americana accession called Winkler, which was used in early Midwest hybrid hazelnut breeding. So what you do, and I don't want to get too far into the weeds here because this isn't really about the technical details of linkage mapping, which is what this is called. This is really about the kind of big picture. How do you use marker data in a selection program to speed things up? But just to mention what you actually are doing on a cursory level, these are all the markers, and the first step of linkage mapping is to arrange these 9,623 insertion deletion markers along the genome. And you do that by measuring the recombination frequency between them, which you can do because of this known population structure, and then inferring how far apart they are based on how many times you observe a recombination event between them. This is a unit of measurement called centimorgans, abbreviated here CM. And this is a very classic technique in quantitative genetics, is to build a linkage map. And then, I have an aside here. We also built a genome assembly, which is just kind of cool. And don't have time to really get into it. For those of you who might know about genome assembly, here are the steps we used. We did long read sequencing of both a Winkler and another important C. americana accession called Rush, which is still used today as a source of EFB resistance. But the point here is essentially that we did this with just a few $1,000s, and I'd never done this before. Building genome assemblies, again, is like all sequencing methods, has gotten both cheaper and easier with time. And the big takeaway here is that the Winkler genome assembly has 97% of the entire genome assembled into 11 chromosomes. So we did I would say we did a great job. This is a really high quality genome that can be used for lots and lots of things other than what I'm showing you here, which is just identifying insertion deletions. This is another busy slide that I don't want to get too far into the weeds on, but this is the next step in linkage mapping, is that you take this map of markers that are arranged across the genome, and then you regress phenotypic data. So that's the Y vector. And in this case, it's a vector of phenotypes. Any one of the phenotypes I mentioned, you can think of that as being plugged into the Y vector here. Percent kernel, length of the nut itself. We do this for all the traits. We build these models where we regress it onto a statistical series of terms that represent what we call additive effects. So that's going to be, there's going to be a term for the additive value of the QTL in parent P, which is Jefferson, and parent Q, which is the UMHDI parent, Eric 421. And then there's also a term in here specifically about what is the contribution of the two of the, is called a dominance term, combining those two specific alleles in this individual plant that we were observing in the F1. So this is how genetic variance, the genetic contribution to a phenotype, is typically decomposed in quantitative genetic analysis, is into these additive and dominance terms, because we care fundamentally about the additive term. We want to know how much variance from Eric 421, for instance, is going to be passed on to all of its offspring, not just when you cross it with Jefferson. And so the point here is really just that we can do this. And that's kind of cool, because that's, you want to be able to make those kinds of statements when you're breeding Eric 421 with lots of potential parents. And then this is what the results look like. So these are plots that are called LOD profiles. It stands for logarithm of the odds. You can think of it as it has a direct relationship with p-value. The dotted line is a representation of what a p-value cutoff of 0.05 would look like with a standard statistical test. Here it's a bit different because of the particular model we're using, and we're making lots of comparisons across the genome. So that's all being controlled for here. Peaks in the profile represent regions of the genome that are highly statistically associated with the trait, which here I'm showing just as an example in shell height. So here are 4 regions, which we call QTL, that are associated with this. And we can estimate the value of the parameters for this trait. The estimates really don't concern yourself with them right now because we don't have time to get into all the details. What matters is that these 4 QTL explain 48% of the variance that we observed in this particular population, which is a lot for a quantitative polygenic trait. None of these individual QTL are explaining that much variance, but together we could predict 50% of the variance in this trait using just 4 molecular markers at this stage in this particular population. Here's in shell shape, length width ratio, where we found 1 QTL that explains only 20% of the variance. Kernel height, I'm going to blow through these kind of quickly for time. I really just want to get to this one slide. Percent kernel gravimetrically. I've kind of been harping on this one because I personally think it's extremely important because it's not just a quality measure, it's a real yield component. And there's 3 QTL associated with it on linkage group, or these are named after the actual chromosomes, 6, 8, and 9. And this again explains not quite 40%, but 37% of the variance in the weight measured percentage kernel, which I think is pretty impressive. This is a real first pass at doing this analysis. So conclusions, polygenic traits, which are sort of all quantitative traits, are going to be polygenic. And these are kind of quintessential traits that follow a quantitative distribution, could and seem to be under the effect of what we would call major effect QTL, regions of the genome, specific, not huge regions of the genome that explain large portions of the variance. And this is promising for developing a marker that you would just sequence that one marker at the seedling stage and say, okay, this is probably going to have a larger or smaller percent kernel. The utility, and we need to do more research here, as always, but specifically, I've got a couple other populations that I'm looking at, and it will be interesting to see if these same QTL are detected in the other 2 families that are very similar to these, same age, they're planted at Rosemount, and so basically, how robust are these specific markers or these specific QTL to the genetic background of the population you're looking at? Maybe they're not, but I hope they are, and we'll be able to test that. Finally, there are other uses of what I'm calling next generation sequencing data, cheap ways of genotyping plants. In particular, you can do what's called genome-wide association. Again, you're looking for QTL, specific regions of the genome that are associated with a trait. This is done in populations where you don't know the population structure, you don't know who the parents were, as well as genomic prediction, which is an entirely different method for trying to not find regions of the genome like genes that are probably associated with some sort of specific molecular pathway that leads to larger kernel fill or something, but instead you just use the markers to estimate the relatedness between all individuals. And this allows you to construct a predictive model that is not explanatory, but is nonetheless can be very, very effective and helpful in applied breeding programs, where using just the genotype, using all of the markers that you have for an individual, so in this case it would be 9,623 markers, you would plug that into a purely statistical model that it uses those markers to figure out how closely related it is to other individuals that you have phenotypes for, and predict its performance. And that's another method that could work very well in breeding programs where oftentimes individuals are very closely related to each other. If you train this model with the parental generation and then use it to predict the performance of the progeny, you can often get pretty high prediction accuracies. I don't have time to talk about that though, but that's the direction we're going in. And hopefully by this time next year, it will be something that Lois et al. Can really use in their research. So yeah, this is all just a bunch of stuff I don't have time to talk about, but I do want to thank the funders, the partners, and in particular, all these people on the right who took a lot of photos of hazelnuts for me. And they're all very esteemed members of the Dawson lab. So Thank you very much.
Thank you, Scott. Devin.
See if I can share. So I do have a hard stop at 2:30 with upper leadership here, so I will, I think 18 minutes will be fine, but just to give you a little bit, I might go through some things a little quicker than others, but I still think I have enough time to, I think, provide some good updates. And it's, I appreciate being part of the larger discussions. It certainly helps me to think about, you know, some of the larger programming that you guys are doing there and within the grant is just within also Forever Green. So let me cue this up, I guess, into presentation mode instead of flipping through the slides. But again, Devin Peterson, I'm really going to talk particularly about one of the students' data today, that being Zoe, who actually just recently graduated. And she focused, you know, quite a bit on what we'll call this sort of what is the non-volatiles that are in your hybrid hazelnuts that are impacting, in this case, flavor quality. And so I hope she doesn't have these in a timer because I sort of stole a few from her slide deck, but if I bounce back and forth, that's why. Okay, let me just actually get rid of some of these windows here. So as I think was already mentioned, certainly a big part of breeding programs such as yours and other genomic, you know, efforts have really focused on the agronomic traits, which makes sense, right? I mean, certainly the goal of the agronomic food supply is to make things affordable and abundant. And I think that, you know, another area that is certainly, you know, a way to consider is, as you're all aware, and I won't spend a lot of time on this because I think it's part of the proposal here, but how do we get what we'll call more consumer traits, you know, as part of our advancements? And so as we're breeding for these needed disease resistance and yield, how do we bring in things that just also are part of how people select food? A big important part of how our dietary pattern is certainly given or impacted by our pleasure of food and what we like. And that is certainly well documented, and I don't need to get into that, but therefore a key consideration if one's trying to think about a competitive product as it relates to both, you know, economic value and also initiatives such as bringing in something more sustainable like locally grown hazelnuts. So I'm going to be really, again, quick on this. I do think that it's always nice when I hear you guys give a little bit of a background of the areas to kind of pull things in. And I think when we think about what is flavor, you know, perception, I'd just like to go really quickly on this, but certainly flavor is, you have three different sensory systems, I guess, in your oral cavity. One of those obviously being olfaction or what you smell. Another one being gustation, which is thought to be about 5 basic taste sensations. I would argue that's an oversimplification, but for today, we're going to go with that. Another one actually called somatosensory. So you have these 3 sensory systems. A somatosensory be, for example, things that would create things like mouthfeel or tactile cues, like astringency, okay, be an example of that. And there's all these have different receptors, you know, olfaction, you probably have 50 million receptors or so in your oral cavity, and the millions also for gustation and alike. And so you have a very complex network of these chemical antennae, if you will, in your oral cavity. And as you're eating, your brain is obviously, you know, or these receptors respond to some of the molecules and not others. And then these patterns are ultimately perceived and are part of what we'll call the flavor experience. And then there's a big part, as I mentioned, of food choice and just flavor quality in general. So what I'm going to focus on is gustation. Another one of my students, who is a very talented student, is working on olfaction components here. I will tell you that historically, most of the weight has been pushed to aroma. People have generally thought aroma is more important. I actually will push back on that, and I think that's a massive oversimplification. The unique thing about gustation is that a lot of things such as bitterness or astringency are more innate to our biology and therefore are hardwired, where aroma is a lot more learned. And if I'm looking at how do I think about advancing, you'd say, flavor quality, innate traits are just have a unique opportunity. I think they're going to have a larger, if you will, hold on what is important for human behavior, as well as just they're easier to model, okay, from a lot of different perspectives. So where I'm going to go here, and again, keep my eye on my watch, is so I don't spend too much time in introduction, but we do a lot of different ways of flavor discovery, and I'm not going to talk about this a lot, but really utilizing, I would call this untargeted sort of chemometrics, flavor omics approach, we'll call it, where the goal is to get really rich chemical data, these samples, and then have them statistically related to sensory outcomes. In this case, we'll say liking or flavor liking. And so, what we do is you guys are kind enough to provide us with as much samples you can of certain cultivars that you've bred. They'll go through a consumer evaluation. In this case, one of the first ones would be acceptance. So we'll use like 100 people. All right, you to get a really understanding of the population, you have to use larger numbers. We'll use it, and so we have to think about how much nuts we have, and we go through and have people actually evaluate them, and we screen for certain types of people even, and there's a lot to this. We then go through and do chemical printing, okay, and to create these really high quality chemical rich say profiles of the different nuts, and then go through a variety of modeling approaches. There's a whole area in my program where we're really getting into different types of modeling, as I'm sure many of you are aware of the machine learning AI type of modeling. But anyways, for this, I'm going to keep this on some of the more our standard approaches. And then we get into this, once we figure out what's predictive, we will then try to understand what those compounds are. Often they are not known. Many times when I define compounds that are very influential for flavor, whether it's in hazelnuts or in coffee, they're often novel. And I'll give you great examples of that today. I'm going to give you 3 new novel compounds that have not been reported in hazelnut. So in this case, we will again go through and identify isolation and allows us to get standards for quantitation, go through isolation or identification, and then actually once we have these sort of compounds, we have to re-add them back or figure out ways to validate their sensory impact. At this point, they're only predictive based on our statistical models, and then we would actually verify that with human behavior. Okay, it's kind of the general footprint of that, or the sort of workflow. And so again, I'm giving you a little bit of very quickly, this is a very standard hedonic scale. I'm not going to dwell on this too much, but we do look at a variety of things. Overall liking is typically how people do this. I also look at a variety of subquality attributes, like if I just smell it, that would be called the aroma liking. If I'm consuming it, I will ask them, okay, just rate the flavor. Because in the essence, things like color and even the texture can have nuances and can have impact, you would say, on how much I like it, okay, because it's an overall liking. When we're doing our work, we really are focused on flavor because, in essence, I'm not probably measuring chromophores or color or things that might have texture, but nonetheless, I'll give you some insight on this in a second. So we really, if I just look at a quick, Pearson's correlation kind of table, at least to let you know, if we look at overall liking scores of these different samples, they really are related to how much they rated the flavor. Okay, this is the highest correlation by far, less to anything else, even less than to just aroma. And what this should tell you is that aroma is only part of the equation. Okay, and it indicates that clearly right here, and that if I have somebody smell them and rate how much I like them, it does not actually correlate only 0.43 to if they actually put it in their mouth and consume it. Okay, so that just gives you some kind of indicator on that. So let me go through again and talk a little bit more about some sensory data. This is again consumer liking data. And so we have these panel, we screen like 5,000 people and we select them based on their dietary patterns or that they are consumers of nut in this case, to get people that are somewhat familiar with this category. One could look at this in different ways. I'm going to make this real simple today. And you see 14, we had 14 hybrids. 2 were actually Europeans that we got from Oregon and were sourced to say, okay, give me some kind of blind control comparison, right, to give us some benchmarks, if you will, of how well are the hybrids comparing to the Europeans that maybe are thought to also be highly successful, right, in that regard, or at least have economic opportunity. And so if you look at this, you certainly see that actually the hybrids are at least in the same, you'd say, statistical space. Some are less light in that regard. But what this allows us to do, I guess, if I'm looking at this, and I can take these now, and I won't dive into this too much, but if we just look at the hybrids, forget about the Europeans, you see changes in liking, statistical changes. And so what that allows me to do now is to do chemical printing and say, what of those chemicals are predictive of the difference in this sort of flavor liking scores, and therefore provide an ability to have that discovery. So, and certainly looking at the data in different ways here. But what I would say is this is just a PCA plot, and everybody knows this is sort of look at how much variation is in your data set. What you want to see, obviously, is the different samples clustering together. These are the different reps, biological reps. And what you will see, of course, is that the Europeans are different than the hybrids. At least they can be differentiated on the second PC, and are also the farthest away in some regards, from on the first PC and from left to right. And so kind of what we had predicted. And so what we do is we took forward we really focused on the hybrids because they are different than the Europeans to create more common, I would say, nut chemistry to sort of have a more, similar kind of material for our modeling. And what we then do is we take our chemical prints. In this case, I don't know where I have that data, but we're probably around 1,000 chemical features. These are sort of predicted to the number of compounds we're actually monitoring the nut. And we'll then model that against, in this case, the flavor liking score. And what this basically shows down here is we can create, very highly predictive models. I mean, that these are high quality models and they're extremely predictive, probably within, within a unit of liking easily. If I was to look at, the overall ability of how much can I predict just if I took a chemical fingerprint, how close could I get to the actual, in this case, liking score? So let me dive a little bit forward here. I want to, again, think about time because I'm going to get lost here in a second on my clock. But what we basically do with this data, and I don't want to dwell on this again a lot. I say this too much, unfortunately, when I'm in a rush. But all these sort of circles represent a different compound. Okay, that's what they are. Forget about this as ESI negative, ESI positive. So what we're doing is we're using the mass spec in different modes because not every mode will see everything. And so we use combinations of mode to get as comprehensive chemical printing as possible. And I guess what I'm really going to focus on this ESI negative. And what you see up here is something. This is a fingerprint of these are what we'll call a way to parameterize the chemical information. So there's a retention time and a mass. And basically what this VIP score is how influential is this compound on the model. And the higher the VIP, the more influential it is. And what it's basically parameterizing is how much variation is in the nuts, as well as how correlated it is, to the, in this case, flavor liking. And this is one way to filter data to say, okay, of all the 1,000 compounds that I'm monitoring, which ones are most predictive of flavor liking? Now, these are negatively correlated. So what does that mean? It basically means is as they increase, flavor liking goes down. So there'd be predictors of flavor disliking. And I might even suggest to you that probably things that affect disliking are more important than things that affect liking. Humans, if you haven't noticed, tend to have a bias for negativity. Just look at the news if you don't believe me. So in the end, people generally focus more on negatives and are more, I would say, avoid negatives more than they seek positives. Maybe is a different way of thinking about this. Not again saying that positives are not important, but if I'm looking at them on a weighted scale, generally a negative is going to be more aversive for behavior. Okay, so I know I have 5 minutes, so let me roll through here. What we end up doing is we take these compounds, in this case, we don't know what they are. I can do the mass spec analysis on them and we know they're unknowns. And so we have to go through a very big painstaking task of taking nuts and purifying them down to single compounds. This is not a trivial feat. And so we go through multiple dimensions, preparative LC, and ultimately to end up with a compound. Why? Because then I take it and do NMR on it, and I also then have a pure standard for quantitation. You can't buy them. We don't know what they are, right? And they're not even commercially available. And so that allows us, in this case, this bandwidth. And I want to show one other thing which is pretty cool actually in this project. So some of the compounds actually turned out to be rotamers. What is basically isomers that are just folded on each other differently. And so we isolated, for example, this is one pair of rotamers. We'll call it compound 5 and 3. And this is when they come out of the nut. They're in this ratio. I apologize. They're in this ratio. You see about 2 to 1. In this regards. And when they're sitting during storage, they start to shift and they start to go back to an equilibrium. And why is this important? Actually, because they both have different behaviors. And so it was quite challenging for us to pull these apart, considering they're rotamers, but we did it actually in a way that enabled us to get some novel discovery here. I'm going to blow through this. Just humor me. We do the identification, and I'm happy to send more details on that. I'm hoping to submit this paper very shortly. Home in on different chemical signatures. Let me get to. I mentioned some of these are rotamers. I'm going to get to the compounds, because we only have a few minutes left. So with this data, what we're able to show here is that this, I guess, phenolic glucoside here is novel, okay, and actually is a bitter compound. These two were actually identified by a prior student who worked within this team on a different project, and is actually was always known to be two compounds, but we can only get it as one. This other sort of compound here, we'll call it 2 and 3, is actually, again, another, this rotamers I talk about, and that they just basically folded differently on themselves. But basically, we have some new discoveries. So we knew one compound before, this one right here, that's also, you'd say, identified, but we identified in this case, compounds 1 and compound 3 and 5, that they are, in fact, I would say, could be classified as different compounds or isomers of each other. So let me just go through some data on this. What is interesting about these compounds, of the top 6 compounds, they all are these sort of indole acidic acid moieties, which of course you guys know is plant hormones. And each one of the top, again, of these 6 compounds that are most aversive, all were built from this hormone and are all the derivatives of this hormone, which I think is interesting. So we quantify them, and let me just give you some sensory data on this. What is interesting about this compound that was the most predictive, if I look at, again, the 12 hybrids and I look at their concentration ranges, the one that's most predictive is, out of all the samples, 91% of the samples had a concentration that was above what's called a sensory threshold for bitter. So it would be saying that this compound would be contributing to bitterness, okay, in those samples. And so we actually had to measure this. This compound is not commercially available. We actually go through and set up panels here and give people amounts of this that have been isolated from a nut. That is, again, just want to stress to you, not a trivial thing. And actually then develop what's called this threshold value, is saying that, okay, anything above this 95 ppm level is now perceived as bitter. And then we can now take that and say, okay, well, what about our nut population? And you could see that this one, again, for 91% of the 12 varietals was above its sensory threshold, indicating it's contributing. If we go to the 2 other compounds that we previously identified, compounds 2 and 4, that were always isomers, what is kind of fun about these 2 is they are slightly different in sensory property. One is a little bit more astringent, one is a little bit more bitter. And actually knowing this is important in regards to, now, if I want to parameterize these compounds, depending on how they're folded in each other, it's going to have a difference in how they're going to be perceived. But again, if you look at these 2, one of them, again, about 83% was above its sensory threshold. The other one is about 25, okay, in these nut samples. And of course, these could, the concentration of these can be obviously influenced by the environment, as well as obviously their genetics. The new ones that we've defined here, again, this indole acidic acid moiety, right, which you see here with these 2 glucose and fructose units, again, showing that these are, I wouldn't say not important, but obviously on a scale, only about 41% of them are above their thresholds. So what, again, we're sort of saying to you is that you have these sort of now molecular markers that are really predictive, or actually causative, of bitter perception, which is a well-known aversive attribute. I know I'm jumping through a lot of things, but bitterness, some people get to like bitter in coffee. Actually, there's some good chemistry on this, or research on this, that's saying because caffeine is the addictive. In fact, caffeine is probably the most addictive molecule in food. In that it is a way that your brain has trained itself to seek out bitter, and that's why you start to like coffee. Actually, there's some good work on that in the literature. But nonetheless, in general, bitterness is aversive. If you give baby bitter, it will spit it out. It will make mad faces, in that regard. And so, anyways, I guess this just gives a little bit. Let me not worry about compound 6. The goal of this is that we have, some new molecular moieties that I think would be great to look at the genomic mapping that was just Scott was talking about, and allowing us to at least provide, I think, a better molecular context to what is driving flavor, in addition to all the other attributes that are being focused on. And that's it. So let me stop there. I probably have about 30 seconds because I know leadership is sitting on Zoom waiting. If there's any quick questions.
How do we move forward on the metabolomics component? How do we connect the chemistry?
Yeah.
So how would we go about organizing that? What type of investment would it take to do that?
So I would say we should have a follow-up call with, whoever would be interested, but Scott and Julian team, and really look at, as you mentioned, how do you take these now and map them back, QTLs or other methods that he mentioned, And then also just for germplasm selection, right? So you can certainly screen these. And now that we have the markers, we can certainly screen them now in the germplasm and probably use that as a way to select
We
germplasm.
probably should bring in Jerry Cohen, right?
Absolutely. Yeah.
Yeah.
What volume of nuts is required for the analysis on each of those?
So now that we have the molecular markers, a couple grams, so a couple nuts. So once it was more of getting these out of the nut in enough quantity, if they're in part per million levels, you could imagine how much I have to isolate of that to get a couple milligrams for NMR. And so that's the painstaking part, is getting the standard right and getting the analytics for the identification of these molecules. Now, certainly one could now start looking at pathways, how they're formed, and start thinking about that in a whole nother complicated way, or add more to this equation. But again, what we're basically showing you here is some really predictive compounds of disliking of nuts, and a way now to minimize those. And it would be my opinion that these would certainly have a positive impact on the flavor quality.
The highest flavor rating was, I think, 6.7. Is that something that you feel good about in a nut?
I do.
Versus like a Snickers bar, or?
Yeah. So a couple things in this. I would, again, if we had more time, that's a very actually good observation from your side. People almost never, when I'm ranking coffee to whatever, never go above 7. Okay, in food products. And so this is a relative perspective, and how comfortable everybody is with their scale, and generally where they scale. So I don't see any flags somehow. And I think having those anchors of those Europeans in there, I hope give you some confidence in regards to at least how these hybrids are relating to those European. I think it was Barcelona and Ennis, if I remember correctly, were the varietals that were chosen. Or
What's the 4 on that scale? The highest one was 6.7. Well, then the bottom was like 4. It's 4 bad.
4 is, if I go to the scale, I'm guessing it's slightly dislike. So 4 would be, dislike slightly. So it's again, a general scale that is quite commonly used in this sort of sensory space, in that regard.
Sean Mellenbacher wants to know how the kernels were prepared for tasting, what kind of roasting time and temperature, and whether the pellicles were on or off.
So I can give you all those details, maybe if you're okay, if you would send me questions, and I'm happy to provide some of that. The roasting was done, and Lois, if you remember, we worked with some of the farmers in your area just to get a couple of protocols from them. And I'm looking at the slide now, and remember, it was 135 for 30 minutes. We tried a few different protocols based on what was in literature, et cetera. But this, at least from our opinion, generated a nice roasted nut flavor profile. And these, by the way, these bitter compounds are endogenous to the nuts. They're not formed during roasting, at least not appreciably. I got to go back and look at the data, but they're certainly endogenous. And the aroma is now another piece we're going to be a little bit more complicated, because there's obviously a reaction step there. But these would be things that you could screen for in the unroasted nut.
And did you take time to remove the pellicles?
So we prepared them. I want to make sure I answer you correctly. I think my student was always connecting with you on how to prepare these. And I guess the question is, was the skin still on it, is what you're asking?
Yeah.
So this is probably 3 years ago, 2 years ago when they did this initial work. Let me get that data before I answer you. For some reason, I'm thinking that it was, I want to be careful what I say. I think the skin was included. But I want to make sure I verify that. And I'm sure that would have been in combination with discussions that I thought.
It comes off naturally with roasting.
Right.
But not always. And that seems to vary by genotype.
So let me dig into that data.
Okay.
And the student's experience.
Thanks, Devin. I'll tell your mother-in-law that you're still doing okay.
I appreciate that. Well, I'm not sure I hit the benchmark yet of success there, but I try. So.
And we'll help on your behalf people that were delayed.
So I will jump up, but I guess if I'm happy, if you want to get another team or whoever would be interested to have a more kind of focused download on what could be next, I'd be happy to do that.
Yeah, I think this is an appropriate time to do that.
Sounds
Thanks.
good.
Thank
Yep.
you.
Thank you, everyone.
There was one other comment slash question directed to me, just to raise up for everybody, especially the Hazelnut diehards. You probably already know this, but there is the first round of grants will be opened up for applications next month for people in the value chain of 4 crops, and hazelnut is one of them. So I think you're aware of that, right? Yep. So anybody on? Say again?
I'm on the edge of my seat.
Good. Anybody on the line, or anyone that you can think of who you could remind of the potential to get, I forget what the numbers are, in the tens of thousands, I think, for the max grant, and buy a piece of equipment, do some market research, whatever it might be that that company needs to do to boost their supply chain. We want people to know about this grant.
Well, we'll make sure it goes up on the website, right?
Yeah.
Make announcements too, and MDA will do the same thing.
So Steve, are you getting all those emails?
Many of them from Colin.
Okay.
And I'm not sure if I'm getting all of them, but I know the core of the chain I'm getting now.
Yeah, there's no official announcement yet either, so we'll make sure it goes to you.
Okay, great. Yeah, I appreciate that.
All right, any other questions?
I answered Don's question from before about the American stuff. So Wisconsin, none of the American that we've had yields anywhere close to as good as the hybrids. Our collection is relatively small. Stuff that's in Wisconsin, and they've looked at a lot more of them, and this is not replicated, but when they're pulling them out from plantings where they have hybrid seedlings and American, the 1 or 2 years that Jason was sending us data from Stoughton, it was like in the top 30, there was 1 American, 2 Americans, something like that. So in terms of performance, it's the hybrids are better.
But I would also argue that we probably shouldn't let that all slide away. Since some of them did rank. And remember, they were being pulled for a while.
No, they're not being cast off, but that's the current state of it.
Yeah. But has anyone made crosses high by high of Americana?
No, they're debating about what they're going to do next with the Americans that they have.
There's a big planting of pure Americana at Hayward, Wisconsin. With seed that was collected from a diversity of locations around.
So
Devin.
it's still in place. It's still.
That was from all that like
Yeah.
original collection.
Yeah.
Yeah, that dates back to.
So has any investment been made in maintaining that and building off of that base?
We're currently trying to decide what to do with it. That's Mike Demchik's
Yeah.
piece, and I probably should have invited him to participate too. He's so busy with his teaching schedule, it's hard to find.
If I could make a comment about that, Lois, I'm no Mike Demchek, but I have looked at the difference between I have tried to look into, what are called the upper Midwest hybrid hazelnuts and what you would classically consider a hybrid, an F1 between pure American and pure Apollona. And it seems to me, is a somewhat challenging question to answer definitively just from sequence data alone and no real pedigrees, but the hybrid hazelnuts are closer to American than they are to European. And that's not, they like the UMHDI varieties. And that shouldn't really, I don't think, be surprising to anybody because of the way those were selected over time was OP selection, where, that pollen cloud is going to be predominantly non-European. And my sense is that what is American hazelnut, Mike Demchek's collection and those diversity panels are pure, unselected, unimproved American hazelnut. And what's considered hybrid hazelnut is in some sense just the improved material, so of course it's better. It's the only material that's experienced selection in the in the 20th century and now. So but
no one has tried to improve and produce hybrids from Americana times Americana.
That's my understanding, is that there is always in the background of upper Midwest hazelnut development has been an impulse to make interspecific hybrids, either with Avellana or with Cornuta. That's always sort of been part of the program. And this, in order to tap into greater genetic diversity, basically. And I don't think there's anything wrong with that. I think that, there are definitely ways of, I think there's definitely, for sure, unexplored improvement. It might not be the most efficient way to go about a breeding program to like go back to a wild species, but that wild species could very well be an interesting crop wild relative for valuable traits. And given the fact that the upper Midwest varieties are more closely related to C. americana than they are to C. avellana, I think including C. americana in training population design and just continuing to look at it and think about it is relevant. It will only improve our ability to make selection.
If I remember that data set correctly, there was great variation within those populations that they were selected from for a wide range of traits, including nut size, if I remember right. So I was always thinking that when that work was started, that one of the approaches that we were going to continue was to advance within the species of Americana.
Those plantings in Wisconsin are at the point now where they can select out of them.
Can they select out of them there? Yeah.
Well, they're old enough, the seed collection, they've made seed collections in the wild and establish these populations.
Yeah.
Rather than trying to cross immediately from the stuff in the wild.
So I would argue that work that had been done should be followed through on, at least to evaluate that material. And see what is now in those selections in straight Americana. David.
I have probably a naive question. So these are trees that from European ancestry, bushes, of course, around here. You're mainly looking at hybrids, F1 hybrids is what your selections are based upon. If you do a back cross between a F1 hybrid and a tree, do you ever get back a tree?
European's not a tree on its own. European's not a tree on its own. It's naturally a larger, taller shrub. It gets pruned into a tree for commercial purposes. There are tree species of hazelnut that are single stem, 80 feet tall kind of things, but that European hazel isn't that.
Can you make that hybrid into a tree though? I mean,
Can we prune it into a tree?
yeah.
Is that what you're
Can you
asking?
turn an American one into a tree?
I've tried and they just keep on producing more suckers.
But if you were to back cross it to.
Yeah.
There are crosses with the tree hazel species that are out there. A lot of them are European hazelnut by Turkish tree hazel, Lois calerna. And they make really Grand Traverse, which was one of the varieties was mentioned, was European times calerna back to European. They make big, huge trees, and we're trying to aim for this sort of shorter, shrubbier, more yield efficient, probably more mechanically harvestable thing from the shrubs. Nobody's been really systematic about trying to like commit to raising the shorter, shrubbier style ones as a single stem, no matter what, take the suckers off however often you have to do it. There is also an interest in grafting our selections onto Turkish tree hazel rootstock because it just does not sucker, but that has other complications that come with it. So, but that's something that's being explored. But mostly we like a lot of the things that the shrub structure brings to it.
Yeah, I mean, from what you were saying, it was going to be part of the garden as well.
The shrub. Not necessarily, because we can drive over them with the mechanical harvester as opposed to the way it's
Pick
done.
up the stems and beat the hell out of them.
Yeah, it just drives down the road. It's the same things are used to harvest blueberries, high density olives, things like that, which is a direction that most tree fruit stuff is growing, almonds, peaches, apples, stuff like that are all. There's attempts to harvest some basics, Oregon's by sweeping off the ground. We're trying not to do that.
Well, we probably should bring this discussion today to a close.
Yes.
Officially.
Mhm. We all go back and forth, yeah, okay, thank you very much to our presenters, and we'll have more about hazelnuts next week, right? All right, thanks, everyone.
Bye-bye. Thanks for everybody.
Thanks. Have a great afternoon.